Hi folks, and welcome to Anatomy and Physiology on Demand. This is week 12, Monday, so lecture 12A. And today we are going to start learning about the central nervous system. And specifically, this topic is going to be an introduction to development of the nervous system. And then we're going to talk about protection that we have in the nervous system. So let's go ahead and get started. Okay, so embryologically, the brain starts developing fairly early, and by week 4, the brain is a linear structure with 3 different bulges. Okay, the bulge that is closest to the tail, in other words, closest to what's going to become the spinal cord, is a swelling known as the rhombencephalon. Cephalon is just a word that means head. Okay. And then going anteriorly to that, we have the mesencephalon. Mesencephalon means middle. And even more anterior, as far anterior as you can go, we have the front brain, which is the prosencephalon. And neurons start developing, and the brain's really developing. These 3 bulges are getting bigger and bigger. But by the 5th week of gestation, this has to change. Now, I know going from week 4 to week 5 doesn't seem like a lot, but in the embryo's lifespan, that's 25% of its life it managed to handle. But by week 5, we now have 5 brain swellings. And what has happened is the most anterior, the prosencephalon, has divided. And the most anterior part is now called the telencephalon. And then the next area is going to be called the diencephalon. The mesencephalon itself has not expanded very much, so it just stays as the middle part of the brain, the mesencephalon. But then the posterior part of the brain also divides into 2 areas, the metencephalon and the myelencephalon. I want to point out this E there in myelencephalon because a lot of students leave that one out. So that's how things are week 5. And as the fetus is developing, the brain keeps on developing, and then we have to make more changes. So by week 13, the telencephalon actually has gotten so big that now it completely surrounds the diencephalon. And so the diencephalon is in the middle of the telencephalon, because the telencephalon is not growing that much, but the diencephalon is not growing that much, but the telencephalon has tremendous growth. And that's at the end of the 1st trimester, beginning of the 2nd trimester. By the end of the 2nd trimester, what has happened is most noticeable in the telencephalon that instead of being smooth, it now has all these hills and valleys. Those words gyri and sulci, which you've learned for lab already. And the whole point of that is to increase the surface area of the brain. And because it's closer to the surface is where we're going to be finding that gray matter full of all those cell bodies. And essentially from that point on until the point of birth, it's just gradually getting bigger and bigger, and we are populating more and more neurons. And so the way the brain looks at birth is pretty much the same way it's going to look in an adult. So if we kind of put this in chart form, and we start on the far right over there, you can see at week 4 we have the 3 swellings, and then by week 5 we have switched them into the 5 swellings. Spinal cord not being part of the brain, so that bottom line, that's where that foramen magnum would be. And these swellings definitely have a correlation to the words of the parts of the brain that we have learned about. Oops, went the wrong direction. The telencephalon basically is everything that we know as the cerebrum. Okay? And that kind of makes sense because when we look at the outside of the brain, that's mostly what we're seeing, the cerebrum with all of its hills and valleys. And then the diencephalon tucked inside, that's mostly thalamus and hypothalamus, and then the epithalamus part. But also coming from here, if you see these little outpouchings on the sides, there actually is neural tissue that's going to go into the eyeball, which is going to form part of the retina for vision. So that's kind of important when we talk about special senses. But for this part, if I ask you what part of the adult brain does the diencephalon become, thalamus and hypothalamus is what I would like to hear back. The mesencephalon is in the middle, and that actually is what we call the midbrain. The metencephalon is interesting. If you go inferior to the midbrain, you know we have the pons with the big pregnant belly. But posterior to the pons, what we have going on over there is the cerebellum. So they are both coming out of the same layer. And then inferior to the pons is the medulla oblongata. So that's what the myelencephalon becomes. So hopefully that makes a little bit more sense now. We've already talked about how the neurons are organized as far as gray matter and white matter. So at this point, if you would like to, I would say pause it and look at each of these pictures, and in your brains, figure out exactly where the gray matter is, where the white matter is, where the cell bodies and dendrites are, and where the axons are located. So coming back, what we're going to talk about now are all the layers that protect the actual brain. And so we're going to start on the far right once again, where we have the most superficial layer. Okay, first we have hair, but the hair is coming out of the skin. Underneath the skin, you have the hypodermis, or the subcutaneous tissue. And underneath that, we have a fascial layer. Remember, we have an aponeurosis that goes all the way from the frontalis muscles to the occipitalis muscle. That's this fascial layer. And that sits directly on the skull bone. So once we have cut through the skull, then we are looking at 3 more layers of tissue before we get to the brain. And those are the 3 layers of the meninges, as you can see on each of the 3 arrows. So we're going to go through those one at a time. Starting with the most superficial one, that's called the dura mater. So mater is the Latin word for mother, and dura is tough. Okay, so it's the kind of a classic anatomy joke for female instructors. On the day we're talking about the dura mater, we all wear a t-shirt saying, I am one dura mater, which is kind of like a polite way of, you know, calling someone the B word. Anyway, the dura mater is not only, okay, so these 3 layers of meninges, let me back up for a second. The 3 layers of meninges are not only around the brain itself, but it's around the entire central nervous system. So around the brain and all the way around the spinal cord as well. The dura mater is slightly different in the vertebral foramen surrounding the spinal cord versus inside the skull. Inside the skull, there are 2 layers to the dura mater. The outer layer of dura mater is actually forming the periosteum of the skull. So it is super glued to compact bone. And then the inner layer actually goes into those fissures of the cerebellum you learned about, okay, like the lateral fissure and the longitudinal fissure. However, surrounding the spinal cord, there's only 1 layer of dura, and it surrounds the whole thing. There is a huge space between the dura and the inside wall of the vertebra, as you saw in lab, and we're going to see again in lecture today. Now, going back to the skull, even though we have 2 layers, for the majority of the brain, those 2 layers are fused together. But in certain places, they are separated, and we call the space between them a sinus. So this is a new definition of the term sinus. The term sinus we've had before is the word sinus in reference to bone, which is an airfield space in a bone. Okay. Now, for the rest of this semester, as well as next semester, whenever you hear the word sinus, not in reference to one of these 4 paranasal sinuses, what that is telling you is this is something like a vein, but it doesn't have the structure of a vein. In other words, it's going to contain venous blood, but a vein has a strict 3-layer structure, and this dura doesn't have that. Okay. So we are going to have some sinuses in the dura mater surrounding the skull, and if you look at the bottom right picture, you can see actually, like for instance, some blue material here and some blue material here, and continuing down there. That's just showing you where these dural sinuses are located. Deep to the dura mater, we have the arachnoid mater, or the spider mother. And so if you look at the top right picture, what you'll notice, there is a complete layer of this, which is the arachnoid mater, from which we have these cobwebby or spider-leggy things that are going deep to it. Okay. So both of these are part of the arachnoid mater, but when we are pointing to a layer, we are usually pointing to the solid layer, which sits external to the spider legs or cobweb, whatever material you want to talk about. The bottom right picture is showing the arrow pointing to the arachnoid mater, and then just below that you could see that there was some dura mater there. So why do I have a picture of a brain here with this whitish covering on it? It's because this whitish covering that you were seeing on this picture, this is arachnoid mater. If you go back to the previous page, what you will notice when the dura mater is present, you actually cannot see the brain tissue. You have to cut through the dura mater to see the underlying brain tissue. Now, if the arachnoid mater is present, not only are you going to see like this glistening little thin layer, you're going to be seeing all the little blood vessels that are external to the surface of the brain or external to the surface of the spinal cord. And the reason for that is they are all tangled up here among the little legs or among the little cobwebs, which are attaching the dura mater to the underlying nervous tissue. The other substance that is in this space is going to be cerebral spinal fluid, which we will come back and talk a lot more about later. So moving on, this picture is showing where part of the arachnoid mater has been removed, and now you clearly can see some sulci and gyri. So do you see the difference between arachnoid mater is present everywhere except the one little area where it has been peeled off and we've lost all the blood vessels and everything else. So that little area in the middle, what you're actually looking at is the surface of the brain, which still has the attached pia mater. Because the pia mater is super glued to the surface of the brain. Okay. It's impossible to peel it off without peeling off neurons. If you try to peel the pia mater off the surface of the brain or off the surface of the spinal cord, it has that really funky edge, like when you see the top of a can of dog food or cat food and it's not perfectly smooth, but it's like all irregular. That's what the surface of the brain would look like if you tried to peel off the pia mater. Okay. And so I have a gross picture of the brain and a histological picture of the spinal cord. And on the spinal cord picture, I have tagged the pia mater, the dura mater, and the arachnoid mater, just so you can see all three of them in relationship to each other. So those are the three layers, okay? The next thing I want to talk about is the ventricular system, okay? So we learned a little bit about this in lab already. We know there are spaces in the brain and they are filled with fluid. And this is the ventricular system, which then leads into something called the central canal, which goes all the way through the spinal cord as well. So when we look at the ventricular system, the first place we start is we look at this big part right here, which on the side view kind of looks like the Starship Enterprise, and anteriorly it looks like the ram's horns. And what you can see is that this ventricle goes from frontal lobe through parietal lobe, all the way back in the occipital lobe, and then has a branch that goes into the temporal lobe. So this can be tagged at any location and it would still be the same ventricle. So we have two of these, one on each side. So we don't call them number one and number two, we call them the lateral ventricles. When you look at a cut surface of the brain, and if you look just below the corpus callosum, you can see that there's this big space right there. So that big space right there is correlating to this area right there, and a lateral ventricle. Now the fluid from there is going to flow down into this place right here, which is exactly in the midline. So this is in the midline between the two oval Easter eggs, the right and the left thalamus. Okay. We have a little area where the right and left thalamus can connect to each other. So that's why it looks like it has a little hole or an eyeball in there. So this right here is the third ventricle, which separates the thalamus, the duck head part, with the beak, the hypothalamus, okay? So thalamus and hypothalamus on each side, third ventricle right in the middle. From the third ventricle, we then have a narrow channel which passes down through the midbrain, called the cerebral aqueduct. It's kind of a misnomer because the midbrain's not part of the cerebrum, but the other name for it is mesencephalic aqueduct, which is what is written down here. But since in lab we gave you cerebral aqueduct because it's a lot easier to spell, I think we're just going to stick with that. Then it continues down to the area between the pons and the cerebellum. Okay. And this area is triangular. And if you put the two triangles together, then you can see that it would be diamond shaped. And so this is the fourth ventricle. Below the pons, you're going to be in the medulla oblongata. So in the medulla oblongata, we're down to a narrow channel. So that is the beginning of the central canal. And then once you're through the foramen magnum, you're going to be in the spinal cord, which still has the central canal going through. Okay, so let's go back to that previous page with the correlations. Okay, the cerebrum from telencephalon also contains the lateral ventricles. The diencephalon, do you remember what that was? Yes, that's right. It was the thalamus and hypothalamus, and the two of them were separated by the third ventricle. Then the mesencephalon was the what? The midbrain. And what's the name of the structure in the midbrain? That's right, it's the cerebral aqueduct. Continuing on below the midbrain, anteriorly we have the pons, and posteriorly we have the cerebellum. Okay, and between the two of them we have a triangular shaped structure known as the fourth ventricle. And then the myelencephalon, which becomes the medulla oblongata, contains the central canal, just like the spinal cord contains the central canal. Okay. So hopefully this makes a lot more sense now as to why things are the way they are. Talking a little bit more about that cerebral spinal fluid. The cerebral spinal fluid is made in a structure known as the choroid plexus. And the choroid plexus is found in each of the ventricles. So it's in the lateral ventricles and the third ventricles and the fourth ventricles. And what it is this really spongy, icky looking mass of capillaries. And oftentimes when we cut brains, whether they're human brains or whether they're sheep brains, students cut it and they think, oh my gosh, when I was cutting it, I must have had something on the blade of the knife because it dumped in there. And so like they're picking out what they think is trash. And actually I'm just going, well, that was the choroid plexus you just threw out. So here's what it is. We've got capillaries. So this is only showing one. So just think of a whole mess of these. We have a capillary extending into the ventricle. So it's completely covered by these ependymal cells, okay? And so what the cerebral spinal fluid is going to be, it's going to be a filtrate of the blood's plasma, which is going to go through the endothelium of the blood vessel and go through the ependymal cells and then end up in the ventricles. So this picture is showing these areas which are red, which basically is just showing the typical location where we can find the choroid plexus in the ventricle. In other words, the entire ventricle is not full of choroid plexus. The entire ventricle is 99% full of cerebral spinal fluid. And there are certain areas where we're going to have the choroid plexus. And this last picture is a coronal section of a human brain. And so these are the two lateral ventricles, and you can see the choroid plexus in the lateral ventricles right there. This right there is that longitudinal fissure superiorly. Okay, so we have something known as a blood CSF barrier. So I put a little review thing up here at the top about the blood-brain barrier, reminding you the perivascular feed contact in the capillaries gives us the tight junctions, and so stuff has to go through cells, not between, to keep stuff out. The blood CSF barrier acts similarly, but it's at the choroid plexus. And now the tight junctions are between adjacent ependymal cells. Where we have the ependyma lining the rest of the ventricles without choroid plexus, there are not tight junctions between those cells. It's only at the choroid plexus do we have these tight junctions. Okay? And so the reason why the rest doesn't have it is because we want to have exchanges between what is in the cerebrospinal fluid and what's in the brain tissue, because we want to have nutrients and pH equilibrium and all that sort of stuff. So at this area coming from the blood, we're going to have a blood CSF barrier. So stuff in this area has to go through the ependymal cells to get into the cerebrospinal fluid. But then the cerebrospinal fluid can easily diffuse stuff back into the brain tissue itself. So the CSF cerebro and cerebro, not cerebral, so it's an O, not an AL. Cerebrospinal fluid that's made in the lateral ventricles will then go into the third ventricle, be joined by new cerebrospinal fluid, and then it will travel through the cerebral aqueduct in the midbrain and goes down to the fourth ventricle, where there's even more being made by the fourth ventricle. And then a lot of it's going to continue all the way down the entire length of the spinal cord. However, at the fourth ventricle, some of it is going to exit. We have a couple of channels where it can exit, and then it goes on the external surface of the brain, the cerebellum and the cerebellum, as well as the external surface of the spinal cord. So it's bathing it not only from the inside, but from the outside. Okay. And so where it is going is between the arachnoid solid layer and the pia. So it's going in the layer where we have all the little spider legs or the cobweb stuff. And that area between the arachnoid mater and the pia mater is called the subarachnoid space. So it kind of goes all the way around in the subarachnoid space. And at certain places in the subarachnoid space, in the arachnoid mater, excuse me, there are like these little mushroom cloudy things that kind of stick upward, which I'm going to talk about on the next slide. And that is where the fluid and the cerebrospinal fluid is going to be dumped back into a sinus filled with venous blood. So the fluid came from blood in the capillary, went into the CSF, and now goes back into the blood in one of these sinuses. So looking at the cerebrospinal fluid itself, it is clear, it is absolutely colorless, okay? There's only about 100 to 150 milliliters circulating at one time, so that is somewhere around a third of a cup. Total fluid around your brain and your spinal cord, plus including inside your brain and your spinal cord. However, your body does make 500 mils of it a day, because remember, we're making it and we're reabsorbing it 24/7. And it flows continuously, not over the pressure of making new, but also because we have those cilia on the ependymal cell. So why do we have CSF? Well, your book talks about 5 or 6 big functions, but I want to narrow it down to the really important ones. The first one is it gives buoyancy to the brain, okay? So the brain is not floating on the surface of this, but it's kind of suspended in it. And so what that does, it takes your brain's weight and it gets rid of about 90% of it. So before you tell me about Dr. Powell, my head feels really heavy. I would tell you that's the bone, not the brain. Students are always amazed when they take the brain out of the skull how much it actually weighs, because you don't realize it in your own head. The second thing the CSF does is it protects. So because it's floating, when you make these little movements like this, your brain is sloshing around in the fluid. Okay, it's not really sloshing, but think microscopic sloshing around in the fluid. So if you turn your head this way, it's not going to go bam and hit that side of your skull. Now, of course, if you're in a car accident, you press on the brake or you miss pressing on the brake and you hit the truck in front of you, your head is going to go bang, bang, and the brain can hit the inside of the frontal bone or the inside of the occipital bone or on the sides. Okay, so you still can get what we call a coup contre coup injury. You don't have to know those words, but basically what that means is hitting this way and then going back and hitting that way, like in a typical whiplash kind of movement. And the third big function is it gives some chemical stability to the brain by regulating the environment, providing it with nutrients so it has a constant steady level, so it's not doing this up and down kind of stuff, and it can get rid of the metabolic waste, which is super important because it does a lot of that while you are sleeping, right? The whole time you're awake, your neurons are work, and they're making waste, and it's building up, and then you go to sleep and the neurons can get rid of a lot of the waste, which is really good. So let's look at that arachnoid villus a little bit closer. So start in the bottom left corner, there's the cerebral cortex and super glued to the pia mater. Then we have that subarachnoid space which contains the cerebral spinal fluid, and then we have the arachnoid mater. So here's that little mushroom that pokes up that I talked to you about. And basically what it is the pressure from here. These are like overlapping shingles. So when more fluid comes in from down below, it just kind of like tilts the shingle open, and then it can go back into the bloodstream. Okay, it's not having to diffuse through anything. There's no valves, there's no gates, there's nothing involved in that. Okay. And so this only occurs in those areas in the skull where the two layers of dura have a space between them in one of those sinuses. And so like we have that big, huge main superior sagittal sinus, which is at the top of the falx cerebra that you could see on the other picture. Okay, so arachnoid villus, another term that we're learning for lecture that we did not have in lab. So this picture is showing you right here at the top of the skull, this is that superior sagittal suture sinus I was talking about. The falx cerebri came down to here. There would be another inferior sagittal sinus. The two of them would both drain. We have sinuses that come along. And do you remember the groove for the sigmoid sinus that we learned about in the bony lamina? So what sits in there is called the sigmoid sinus. And so by the time it has reached a sigmoid sinus, everything from these sagittal sinuses has drained and has all coalesced. And now those two sigmoid sinuses are taking it directly to the jugular foramen, and on the other side of the jugular foramen, we then call it the internal jugular vein, because now it's a vein. Now it has the three layers that veins are supposed to have. And so that's how the venous blood exits from inside the skull. We have put all the pieces together finally. And with that, I'm done with this first tape. So thank you for all your hard work, and I will see you again shortly for the next one.